the purpose identified, then we know under different conditions, physiological conditions, pathological conditions, these cells, number of these cells vary. Number of these cells vary. এটার পেছনে একটা ইয়ে আছে না, বোর্ড আছে না? The case of neutrophil, we could identify the neutrophils. Then theoretically we know that neutrophils may have different lobules of nucleus. Nucleus may be lobed. The young ones will have two, and when their age will be increasing, the number of lobes will be increased. And these lobes are not separated from each other. They are connected to each other by a thin fiber-like structure. They are not completely separate. So the neutrophils are lobulated. They are having different numbers of lobes according to their age. Having 6 lobes means they are elderly. And now it is their time to die. And when they will die, what will happen to them? One is when a cell dies, either in blood, a blood cell dies in blood or in tissue, a cell dies, then what will happen to it? What will happen to it? Tell. You know that I in my class repeatedly tell this. It happens like that.
Destroyed cellular transport to the spleen and
since they are dead, they cannot remain in circulation. So they will be trapped by macrophages, or they will be phagocytosed. When they will be passing through the liver or passing through the spleen, they will be trapped and they will be metabolized. Because when they will be phagocytosed, the cell, the dead cell will be having chemical constituents like these are made up of protein, fat, this thing. So they will be splitted, digested there by the enzymes present in the lysosomes. Isn't it? That's the way they will be removed. There are different kinds of neutrophils. Not kinds. We have differentiated them, classified them according to the number of lobes. Okay, let's see. We have studied 100 neutrophils. How many? 100 neutrophils. To paper. To board to nahi ekhane. Paper dao. Some looked. I'll be studying 100 numbers of what? Neutrophil. So out of this 100, some will be having 2 numbers, some will be having 3 numbers of lobes, some will be having 4, and some will be having 6, 5 and 6. Let's see their number. I have studied how many? 100. So out of this 100, my 4 lobe is 40. When I so what I did, I saw 1, then I saw 2, then I saw another 1, 4, then I saw 2. So this is 1 across it. 1, then 1, 2. So in this way I will be counting. And after that, total number will be 100. Okay. Now see, out of this 100, how many are having 2? How many are having 3? Total, how much is this? 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 Now with this, we can plot a graph. How? In x-axis. Which one is x? This one. This one x.
No, sir. This
Sir, why?
this one, sir.
I wanted to confuse you. So this is x and this is yx. Y axis. Now, these are the numbers. Percentage 10%, 20%, 30%, 40%, 50%, 60%, 70%, and so on. I have calculated, and here is 2-lobed, here is 3-lobed, here is 4-lobed, here is 5-lobed, and here is 6 and above. Is that okay? Now, put this value according to the scale. Suppose 2 is 10%, 6 is also near about 22%, and 4 is 40%. This is 40%, isn't it? The 40%, and 3 is here, 4 is here. Okay. Now we can join this. Out of this, these numbers, percentage of different lobe neutrophils are called Arnett index. ARNETH. Arnett index. So when we will be drawing this curve, we can see this number is maximum, and their number newly formed neutrophils are minimum, and older one are also minimum. That means number of neutrophils young produced, it is same as the number of neutrophils dying. That is, neutrophil production are in a state of homeostasis. Homeostasis, balanced condition. Okay, is balanced. It indicates I am neither, I don't have any disease which can reflect neutrophil, and if the curve is like this, newly formed is zyada, more. Means some infection, some outsider has invaded in my body, so my defense system started reacting to this, producing this to combat with the invader. That's why its number is rapidly rising. So with this index or through this curve, we can predict whether I have been infected or not, or there is any invasion of in which the cells are reacted. Okay, and if the curve is like this, that's opposite. Newly produced neutrophils are less, and older one is more. That means New neutrophils are not producing. Something may happen in my bone marrow. Why it is not producing? So there is something wrong. So with studying this neutrophil, we can estimate, we can predict whether something wrong in hemopoiesis or is there. Whether it has been invaded or the function from where it is, or it may die also. After newly formed, it may some agents are killing them. It may also happen. So it is not normal. It's not normal. So we can, that's why this is the importance of RNA index. Could you follow? Okay. Then this is one purpose. And another purpose is like this, not only neutrophil, say he became allergic. So there will be response. Those blood cells which fight against allergy, their number will be increased. Example, which cells? Basophil, eosinophil. Their numbers will be raised. So, you are allergic. So, let's see whether it is reflected in the blood picture. So, I have to study this. We have to confirm this. So, for this confirmation, or as a virus has infected me, or a bacteria has infected me. So now, since these are foreign bodies, so immediately there will be changes because they are involved with my protection. So let's see whether it is adu, whether it is at all fighting with this process or involved themselves with the process. So if they are involved, their amount will rise, their number will rise. So initially, before we count the number of RBC, number of WBC, number of platelets in the blood, number of WBC, different WBCs. So this is the total count of, and that the procedure is different. Now what we can do with this blood smear? We can do another, like RNA index, we can do another test that is differential count of WBC. WBC are different types. So we will study here also 100 WBCs. All the WBC, there will be all types of WBC. Let's see. What are the numbers of neutrophil? What are the, out of this 100, how many are neutrophil, how many are lymphocytes, how many are monocytes, okay? How many are basophils? So this number out of 100 is called differential count of each variety. Okay, so after making the smear, staining it, drying it, you have to study this under microscope. And first you have to recognize them, identify them. Then your part is to go for differential count. How will you do it? It's like this only. Instead of this 2, we'll write here N means neutrophil, E for eosinophil, B for basophil, L for lymphocyte, M for monocytes. Total number how many? 100. So now say how could you, out of this 100, how many neutrophils you could see? So if it is 60, then it is 60%. If you identified one basophil, only one, or may not you get out of 100, there may be absence of any basophil. So that is 1%. If you get one, then it is 1%, and it is 60%. And if you get eosinophil 6, then it is 6%. And if you get monocyte, what is the percentage of monocyte in our blood?
20 to 40%.
20?
40%.
40%. Lymphocytes. No, monocytes.
Monocyte to absolute.
Monocytes value? Lymphocytes value? Normal value? It is 25% approximately monocytes. The rest will be definitely monocytes. So this is, if you count the WBCs this way, so this counting WBCs in the way is called differential count. In different viral disease or in tuberculosis, cancer, these are going to be changed. Picture differential count or this number will be varied. So with that you can confirm or you can suggest or you can diagnose, yes, this sort of infection is there, viral infection or bacterial infection is there. So this is the purpose of doing blood smear. And another importance of blood smear is to find out any abnormal cells that are not abnormal blood cells. This RBC looks big, almost the size of WBCs. Then it is macrocytic. In anemia it happens. Okay. Some abnormal cells you can also get there. Maybe a cancer cell. So for identification of abnormal cells also blood smear is needed. So this is the importance of blood smear. Now sir will tell you how to prepare the blood smear.